Welcome to The Explainer. Today we're diving into something that honestly sounds like it was pulled straight out of a science fiction novel. We're talking about a massive, radical shift in how we might treat one of the most stubborn diseases in human history, cancer. For decades, the absolute gold standard for fighting cancer has been systemic chemotherapy. And it works, but it's brutal. It's an exhausting process that often feels like using a sledgehammer to fix a tiny, delicate watch. But what if we could just put the sledgehammer down? What if, instead of flooding the entire body with highly toxic chemicals, we could guide a microscopic payload directly into a tumor using the invisible, completely undeniable force of magnetic physics? That's exactly what we're looking at today. It's called magnetic drug targeting, and it's a cross-disciplinary breakthrough that could genuinely change the future of medicine.
To really wrap our heads around why this is such a giant leap forward, we kind of have to rethink our basic approach to medicine. You see, most modern, smart cancer drugs are fundamentally biological. They rely on these tiny biological handshakes, using things like antibodies to search the body and bind to very specific receptors on the surface of a cancer cell. And hey, it's incredibly clever. But biological handshakes are, well, they're fickle. Cancer cells mutate constantly, receptors change, and the environment inside a tumor is just chaotic. So researchers asked a genuinely brilliant question. What if the key to beating cancer isn't better biology, but better physics? What if we bypass all those incredibly complex biological handshakes entirely and instead just rely on raw, predictable physical force to put the drug exactly where we want it?
And the need for this new approach, it absolutely couldn't be more urgent. Let's just ground ourselves in the sheer scale of the problem for a second. In the year 2012 alone, there were about 1.3 million cancer deaths just in the European Union. 1.3 million. That is a staggering number, and it tells us something really crucial. Our current methods, despite all the billions in research and all our advancements, have hit a hard clinical ceiling. You see, the effectiveness of current cancer drugs is directly tied to the actual concentration that makes it into the tumor, but because these drugs are inherently toxic, we hit a physical limit. There's an absolute maximum amount of toxic drug a human body can process before experiencing total systemic failure.
Which brings us right to the core problem with systemic chemotherapy. Like we said earlier, it's the ultimate sledgehammer. Because chemo is usually injected intravenously, it circulates indiscriminately throughout your entire body. It ravages healthy tissue right alongside the malignant cells. We're all familiar with the profound, devastating toll this systemic burden takes on a patient. Magnetic drug targeting, on the other hand, well, it acts like a sniper. It's a precise, highly localized approach that completely flips the physical distribution of the medicine. The whole goal here is to fundamentally transform a systemic poison into a highly targeted, localized precision strike.
Okay, so you're probably wondering, how do we actually pull off this sniper effect? It all comes down to a funny little acronym, SPIONs. That stands for Superparamagnetic Iron Oxide Nanoparticles. Basically, you can think of SPIONs as these microscopic, custom-built delivery vehicles. Right at their center is an iron oxide core, which, crucially, is magnetic. Surrounding that core is a safe, biocompatible shell, and attached to that shell is the actual chemotherapeutic drug payload. A common one is middlefantrone, for example. Just think about how wild this is for a second. These SPIONs are so unimaginably tiny, we have to measure them in nanometers. Yet, because of that magnetic iron core, we can actually steer them and control them from the outside using external magnetic forces.
Let's look at how this delivery actually plays out in practice. Instead of your standard IV injection that just dilutes into the bloodstream and gets filtered out by the liver, the researchers use an intra-arterial approach. That means they inject the SPIONs directly into the specific artery that's supplying blood to the tumor. But here is the absolute genius part. As the rushing blood carries these particles right toward the tumor, they place an incredibly powerful external magnet directly over the tumor site. The magnetic field gradient literally yanks the nanoparticles out of the rushing blood and traps them deep inside the tumor tissue. So the drug gets delivered exactly where it needs to be and absolutely nowhere else. Now, the results of doing this, they're nothing short of astounding. When researchers tested this in preclinical models, specifically using New Zealand white rabbits that had really aggressive squamous cell carcinomas, they achieved what a lot of people consider a holy grail in nanomedicine. They were able to get a complete remission rate using merely 10% of the normal toxic systemic dose. 10%. Let that efficiency sink in for a second. Just 1/10 of the usual chemo payload was needed, simply because 100% of it was being trapped exactly where the cancer lived. The entire rest of the body was completely spared that heavy toxic burden, while the tumor took a massive, concentrated, direct hit.
you can actually see the undeniable proof right here on the screen. Take a look at these 3D angiographic scans of the rabbit's leg. In the before image on the left, you can see a massive, highly vascularized tumor mass just completely taking over the limb. Now, compare that to the image on the right. That was taken just 11 weeks after the magnetic drug targeting treatment. The tumor is completely gone. I mean, even the abnormal blood vessels that were feeding the tumor have completely vanished. And in the 120-day follow-up, zero recurrence, no metastases. It is just undeniable visual proof of complete tumor destruction. But you know, the journey to getting these amazing results wasn't exactly smooth sailing. In fact, for a long time, the early physics models insisted that this kind of result was practically impossible. When scientists ran the early computer simulations, the in silico models, the math basically said the hydrodynamic drag from the blood rushing through the arteries would just be way too strong. The computers said the blood flow would wash all those tiny magnetic particles right away. No way a magnet could hold them. But when they actually did the real-world in vivo experiments, the particles did get trapped. It turns out the math was just missing a crucial piece of the puzzle. It was likely the complex magnetic interactions that happen between the particles themselves once they start grouping together. Basically, reality completely defied the simulations. And believe it or not, that wasn't even the only bizarre plot twist in this story. As researchers were trying to run standard safety tests on these new nanoparticles, they stumbled right into a massive blind spot in medical testing. You see, the absolute standard toxicity test in biology, it's called the MTT test, relies heavily on light. It measures absorbance or fluorescence to tell if cells are healthy or dying. But here is a completely wild, unforeseen fact. Because these SPIONs are made of iron, they are optically active. They physically absorb and scatter light, so they completely blinded the traditional tests. The machines were just spitting out false positives and false negatives left and right. The team literally had to innovate entirely new safety protocols, eventually shifting to something called real-time cell analysis, or RTCA. This new method uses electrical impedance instead of light to measure cell health, totally bypassing that optical interference. Just brilliant problem solving. Of course, having a great idea in the lab is pretty much useless if you can't fit the actual equipment into a hospital room, right? Back in 1999, the electromagnet you needed to generate a field strong enough to trap these particles was an absolute behemoth. We're talking 1,500 kilograms. You certainly couldn't just wheel that thing into an operating theater. But check out the engineering marvel that has taken place since then. Today, engineers have successfully shrunk that massive machine down to a highly mobile, 3D adjustable unit that weighs just 47 kilograms. And astonishingly, they did all this shrinking without losing any power. This nimble little machine still generates a staggering 1 Tesla magnetic field to pull those nanoparticles right out of the blood. And this compact technology unlocks something that sounds, again, straight out of a sci-fi movie. It's a concept called theranostics. It's exactly what it sounds like, the fusion of therapy and diagnostics right into a single platform. And it works all because of a brilliant, totally accidental coincidence in physics. Because the SPIONs are made of iron oxide, they naturally double as perfect contrast agents for MRI scanners. This means that as the patient is actively receiving their cancer therapy, doctors can watch it happen in real time. They get this incredible God's eye view of the magnetic drug physically accumulating right inside the tumor. They don't have to wait weeks to see if the drug actually hit its target. They know instantly. And that allows them to adjust the magnetic field on the fly to perfectly paint the tumor with the drug. Which brings us to our ultimate question. By stepping totally outside the boundaries of traditional biology and teaming up with physicists, engineers, and even mathematicians, these researchers have built a platform that just defies the old limits. We've seen how a 10% dose can wipe out an entire tumor, how 1,500-kilogram magnets became mobile clinical tools, and how doctors can actually watch a drug destroy a tumor in real time. So what does this all mean? Are we finally witnessing the end of Sledgehammer oncology? If we can guide our absolute most potent medicines with the invisible pinpoint accuracy of a magnetic compass, it just might mean that the future of treating disease won't just be about inventing better drugs, but about having better aim. I hope this explainer has left you as completely fascinated by the potential of nanomedicine as I am, and really eager to see exactly where this incredible physics-based approach takes us next.